Hi everyone. This is the next lecture of our discussion of specialized receptors or specialized senses. And today we're going to cover lecture 15, olfaction, the sense of smell, and gustation, taste. So here we have the nasal epithelium, and in this picture we have the tongue to indicate our focus for today. And pretty much we're going to, for both, we're going to cover the receptors of each and their neural pathways, and briefly describe, how each receptor is uniquely evolved for their specific function. We're going to find that there's going to be some similarities between the other special senses we've discussed and some differences. So let's start with olfaction first, which is the physiology of smell. Of course, we're going to talk more about the area of the nose. And olfaction is the detection of odorants or chemicals in the air that transduces signals to the brain that they can interpret. So when we smell something, even though it's something we say is out in the air or in the room, it really is those odorants that are inhaled, dissolving into the fluid of the nasal epithelium, binding to the receptors to let our brain know that something smells good or smells bad. Okay, so when next time you smell something bad, it's a little more disgusting because it's actually now part of you. But we still say it smells bad out here. So let's look at the structures of olfaction, and our primary focus will be on the olfactory epithelium. And we kind of know about this already a little bit because you learned about the bones of the skull in the nasal cavity. So the olfactory epithelium is going to be in the nasal cavity, and here are showing the nasal concha, and you learn in SCB 203 that the subpyramidal nasal concha are part of the ethmoid bone. And here's the cribriform plate of the ethmoid bone, and you learn that there were little holes in the cribriform plate called the olfactory foramina, and these are the allows the free nerve endings of the olfactory neurons to detect odorants and send these messages to the olfactory tract in the olfactory bulb and to the brain. The olfactory epithelium has three types of cells: olfactory neurons, supporting cells, and basal cells. Here I made a brief table indicating each cell and their functions. The olfactory neurons contain the chemoreceptors, so they're the ones responsible for detecting the odorants. We also have supporting cells, and these are pigmented cells that support olfactory neurons. And finally, we have basal cells. These are the stem cells that produce more olfactory neurons. So already we see something unique about the olfaction, is that the olfactory neurons, they regenerate. We have about 10 million olfactory neurons. This allows us to detect a wide array of odorants. So here in this diagram, it shows the olfactory epithelium. Here you can see the cribriform plate of the ethmoid bone and the olfactory foramina, showing the olfactory neurons as they extend through the little holes. Each dendrite of the olfactory neuron has these non-motile olfactory cilia that project into the mucus from the olfactory glands. So here would be the mucus out here, and you can see here's the dendrite and the olfactory cilia that's extending out. So odorants from here would bind to the receptors on the cilia. And we can see olfactory glands here that are going to help secrete the mucus and the fluid lining the olfactory epithelium that will help trap odorants. Here's an electron micrograph showing the dendrites of the olfactory epithelium, and you can see the little cilia sticking out. They sort of look like little daisies. Now, as you recall, the axons pass through the holes of the cribriform plate, form the olfactory nerve, or cranial nerve 1. And the axons synapse in the olfactory bulb form the olfactory tract. Okay. So here's the olfactory bulb out here. Here's the olfactory tract, and these are formed from the olfactory nerve. And as a synapse with the specialized olfactory neurons. Next, we also have supporting cells. And these are pigmented columnar cells that support and surround the olfactory neurons. I'm not quite sure what the purpose of the pigmentation is. That I cannot find out. So here are the supporting cells in purple. So you can see there's quite a few of these supporting cells. And you can see that they're in a very good structural location to help protect and support the olfactory neurons. Finally, the third type of cell are basal cells. And basal cells are stem cells that develop the olfactory neurons. So here, you can see they're less frequent. In blue are the basal cells. And olfactory neurons have a lifespan of about 30 to 60 days. And this is very unique because most neurons do not regenerate. So in the olfaction, the olfactory neurons are able to replace themselves through these basal cells. So this is one unique feature of olfaction. Let's look at part of the physiology of olfaction. So the function of these olfactory neurons is to detect odorants and send neural signals to the brain. So we can smell, have the different sensations of the smells. So the odorants dissolve the mucus surrounding the olfactory epithelium. And they dissolve in the mucus and bind odorant binding proteins to receptors on the olfactory cilia. And these receptors activate G protein receptors, much like the G protein receptors we saw in vision. And when they're activated, these G proteins separate from the receptor. Okay, so this picture is much like we saw in vision. Here's a receptor. When it's activated here by the odorant, the G protein separates, and here it binds. Well, we'll see it binds to an enzyme. The activated G protein triggers an enzyme called adenylate cyclase to convert ATP, which we know already, into a structure called cyclic AMP. So this is a very similar structure to cyclic GMP we saw in vision. So here in this part of the diagram, we see the activated G protein leaving its receptor, binding to the enzyme called adenylate cyclase to convert ATP into cyclic AMP. Cyclic AMP is a second messenger. And what it does, it opens ion channels that will allow sodium and calcium to enter the cell. As these are both cations, their entrance into the cell will cause depolarization of the olfactory neurons. So we can see in this last part of the diagram, cyclic AMP binds to the channel and opens the channel to allow sodium and calcium to depolarize the membrane. Remember, cyclic AMP is an example of a second messenger. And one of the advantages of a second messenger is that it amplifies the signal. So one odorant binding to the receptor will cause an activated G protein to bind to an enzyme, but instead of one cyclic AMP being formed, we may have thousands of cyclic AMP being formed, binding to many of these channels to depolarize the membrane. So once the neural signals enters the central nervous system, it needs to be it is then detected to identify an odor. Okay. So the axons that carry stimuli to the olfactory bulb in the central nervous system. These axons synapse with dendrites of the mitral cells in the olfactory bulb. Okay. So these cells here are called mitral cells. And the axons of the olfactory neurons synapse with these mitral cells in the olfactory bulb to form the olfactory tract. And as you saw that in pictures of the brain from the laboratory, you saw here this is large olfactory bulb on the ventral side of the brain. So the stimulus travels from the olfactory bulb to the primary olfactory cortex in the temporal lobe. Okay, the primary olfactory cortex is in the temporal lobe. Now, the olfactory pathway is unique from others that we've studied so far, or we will study, in that the sensory pathway has no synapse in the thalamus. Okay, we mentioned that the thalamus is like the grand central station of the brain, that pretty much everything goes through the thalamus. One exception, however, is the olfactory pathway, where there is no synapse in the thalamus. And this diagram is now showing the primary olfactory cortex in the temporal lobe, indicated here and here. Now, the olfactory pathway, however, is very interesting because it contacts neurons in many different parts of the brain, such as the amygdala, hippocampus, the hypothalamus, and other parts of the limbic system. It also makes connections to the frontal lobe to integrate olfaction and taste. Now, you might ask, why is so many, what's the purpose of all these innervations? Well, let's think about it. Suppose you you smell baked cookies. In your amygdala, that might raise an emotional sense that you miss your grandmother, who used to bake the cookies. And this memory is then invoked by the hippocampus. Other parts of the frontal lobe may allow you to remember the recipe for the cookies. So this just gives some example of how sort of elaborate the olfactory pathway it is to innervate different parts of the brain. Okay, now let's go to taste. And we'll cover the anatomy, and then we cover the physiology of taste, which is clinically termed gustation. And just like all the other special senses, it requires receptors, and here it requires chemoreceptors. And these are on specialized cells called taste buds. And so here's your tongue, and it's covered by projections called papilla. And we have 4 types of papilla based on shape. Okay, so here are the 4 types of papilla. We have valate or circumvallate papilla. We have fungiform papilla. We have foliate papilla and filiform papilla. Let's look at the valate circumvallate papilla. These are the largest, and they're dome-shaped. Okay, and so here's the valate papilla, and they contain hundreds of taste buds. The fungiform papilla are named because they're mushroom-shaped. And here are the fungiform papilla, and they contain only a few taste buds. So not all the papilla are the same functionally. The foliate papilla are on the ridges on the side of the tongue, and they only contain taste buds in childhood. Finally, the filiform papilla, which is not shown in this figure, they're scattered across the tongue, and they have no taste buds, but are important for the texture and temperature of food, and detecting the texture and temperature of food. Okay, so now we know the different structures that taste buds may be found on. Let's talk a little more about taste buds. Okay, we're born with about 10,000 taste buds, but we lose about half by adulthood, and this declines about 50 years of age. So, like, I remember my father, you know, he would add more salt and more seasoning as he got older because he was losing his taste buds. Taste buds are located on the lateral surface of papilla with 3 cell types. We have gustatory cells, we have basal cells, and we have supporting cells. So a little similar to the olfaction. Let's talk about the gustatory cells. They have a lifespan of about 10 to 14 days. So they regenerate as well. There are about 100 epithelial gustatory or taste cells on taste buds. Okay, so here are these gustatory cells in orange here. The microvilli on apical surfaces receptors that detect different tastes. Okay. And the microvilli projects into a taste pore. So here's the taste pore, and you can see the microvilli sticking out. The sensory neurons synapse in the CNS via the facial, glossopharyngeal, and vagus nerves, which are cranial 7, 9, and 10. The basal cells are the stem cells that produce new gustatory cells. Okay, so here in blue are the basal cells. The supporting cells provide physical support to the gustatory cells, and so here purple are the supporting cells. So let's talk a little bit about the physiology of gustation. Taste involves transduction of food chemicals into neural signals. So generally we have several different taste sensations, and it continues on the next slide. We have sweet, sour, and salty here. And here in the right-hand column shows the source of the chemical that is used to detect these sensations. So sweet will be simple sugars like glucose. Sour will be hydrogen ions like in citric acid. Salty will be produced by metal ions like sodium and potassium ions. We have bitter, which is produced by nitrogen-containing compounds or even rancid or poisonous substances that kind of tell us that something might not be very good because it tastes bad, and that's why most medicines have these bitter tastes, so we don't actually take them. And finally, umami, which is a taste associated with meat or broth, and this is due to glutamate or other amino acids. Okay, so just like in olfaction, taste is detected when stimuli bind receptors on the microvilli of gustatory cells. One type of receptor is found on each gustatory cell. So despite what I used to learn when I was in high school, different tastes are not localized to separate regions on the tongue. So I remember in health class we would see a tongue and we'd have different sensations on different parts of the tongue, but that's no longer known to be true. A substance dissolves in saliva to reach the gustatory cell and transduce into a neural signal. Okay? And then as a result, the gustatory depolarizes by movement of ions. And depending on which sensation it is, it will be due to a different mechanism. So if it's salty, it'll be sodium ions. If it's acidic, it'll be hydrogen ions. Okay? It may activate G proteins, or indirectly it may close potassium leak channels for other tastes. So in terms of gustation, there's many different mechanisms by which the different tastes can be known. Okay, so here's an example of showing here's ions like sodium or hydrogen ions entering the cell to depolarize the membrane. Okay. And these open voltage gated calcium channels due to depolarization. Okay, this is nothing new. However, in this case, action potentials do not occur. So here's something different about gustation. Gustatory cells are small enough that local potentials are sufficient. Okay, so here's the calcium channels, the voltage calcium channels there will depolarize the membrane, which will cause neurotransmitter release. So calcium triggers neurotransmitter release, producing action potential. And there are a variety of neurotransmitters, in addition to the variety of mechanisms of activation. So there's serotonin, glutamate, acetylcholine, and even gamma-aminobutyric acid can be used, or GABA can be used as a neurotransmitter. Okay. So depending on the type of neurotransmitter, upon depolarization and production, depolarization of the membrane, synaptic vesicles are releasing the neurotransmitter. And these bind to receptors on the sensory neuron to then stimulate an action potential. As we mentioned, the gustatory pathway involves 3 different cranial nerves. The facial, glossopharyngeal, and vagus nerves all carry stimuli to the central system. And here is showing the 3 different cranial nerves as they send their message to the central nervous system. Okay. And so, in the previous figure, I skipped it, but we'll look at it here. And this compares the 5 major special senses. We have gustation, we have vestibular sensation, hearing, vision, taste or gustation, smell or olfaction. Okay. Some things that we can see immediately is that olfaction is the only one that does not synapse in the thalamus. Okay. And it goes to the temporal lobe. However, taste, hearing, and vestibular sensation, they all synapse in the brainstem. Okay. But the optic vision does not. However, all 4 of these, gustation, vision, hearing, and vestibular sensation, all synapse in the thalamus. So we said that the olfaction has its sensory, primary sensory area in the temporal lobe. Okay, taste is in the parietal lobe. We know vision is in the occipital lobe. Hearing is also in the temporal lobe, and vestibular sensation is in the parietal lobe. Okay, and both of these have innervations to the frontal lobe. So this allows you to notice that the air smells a particular way. Okay. This allows to incorporate vision and also the details of what's occurring. So here you can see that the air smells salty, for example, that you realize the wave is big, and you see your friend that's falling off his surfboard. In the limbic system, you can remember that this is the beach where your family vacations each year. It allows you to recognize your friend, and it allows you to have compassion that you're relieved that your friend is safe. So, so these are some of the complex mechanisms that can be involved in how these different senses are interwoven into the different parts of the central nervous system. So hopefully this provides an introduction to the complexities that are involved in the special senses other than their primary sensory function.